My name is Swastik and I am going to present on the topic photorespiration. So what is photorespiration? It refers to a process in plant metabolism where the enzyme rubisco oxygenates RUBP, wasting some of the energy produced by photosynthesis. It can also be defined as an attempt to reduce the consequences of a wasteful oxygenation reaction by enzyme rubisco, or the process that leads to only one 3-PGA or phosphoglyceric acid being produced in the dark reactions in chloroplast. So it all starts with the enzyme rubisco. Rubisco has two types of activities: carboxylase activity and oxygenase activity. The carboxylase activity results in Calvin cycle, as we all know, and the oxygenase activity results in photorespiration. So in carboxylase activity, RUBP combined with carbon dioxide in presence of rubisco form two PGA molecules. These PGA molecules are then converted to RUBP, releasing sugar. But in oxygenase activity of rubisco, RUBP combined with oxygen produce PGA plus PG, which is phosphoglycolate. This phosphoglycolate cannot be used in Calvin cycle, so they are transformed into PGA molecules. This process, transforming PG to PGA, is called photorespiration. So this process is called photorespiration, and in this process CO2 and ammonia is released and ATP is consumed, as you can see in this graph. Photorespiration in plants. If the stomata is closed, the oxygen from photosynthesis will build up inside the leaf while the carbon dioxide will not get into the leaf. This situation will make the concentration of oxygen inside the leaf higher than the carbon dioxide, and the rubisco will more likely bind to the oxygen. This is why the photorespiration happens. Photorespiration depends on the light and also the amount of oxygen and carbon dioxide in the atmosphere, which is favored by high oxygen and low carbon dioxide. Talking about oxygen, photorespiration rate decreases linearly with lowering of atmospheric oxygen concentration because it is required for producing glycolate in the process of photorespiration. Also, with increased temperature, at higher temperature rubisco is less able to discriminate between CO2 and O2. This is because the N-adiol intermediate is less stable. Increasing temperature also reduces the solubility of CO2, thus reducing the concentration of CO2 relative to O2. So what is the purpose of photorespiration? Reducing photorespiration may not result in increased growth rates for plants. Photorespiration may be necessary for assimilation of nitrates from soil, but it has been postulated that from photorespiration may function as a safety valve, preventing the excess of reductive potential coming from an over-reduced NADPH pool from reacting with oxygen and producing free radicals, as these can damage the metabolic functions of the cell by subsequent oxidation of membrane lipids, proteins, or nucleotides. Thank you.